In our previous video, we introduced you to the first of the four classes of amino acids, that is the nonpolar amino acids, and we covered 9 of those, so that's by far our largest group. On the next page, we want to go to the next group, which is the polar amino acids, and in particular, we're interested in the polar neutral amino acids. That is, those that are polar but are not charged. And now let me make sure you understand, of course, all amino acids are charged. They're always going to have a negative and a positive charge to make them a Zwitter ion. So when I say neutral and polar, I mean simply the side chain. So let's get started. The first one we want to talk about is serine. And serine has an alpha and a beta carbon. And it's very much like an alanine in that way. But we now have an alcohol attached to it. And it's a very simple, small, and common amino acid, and it's one of the easiest to draw. Threonine, by the way, is very much like serine. It's almost identical, but it does have this extra methyl group attached to the beta carbon. So it's like an alanine with an alcohol and a methyl group attached to it. Now, I usually think about these two as part of the same group because they're so similar to each other. They are good for hydrogen bond donor or acceptor, and they can interact with water. And because of that, they tend to make proteins that have them be slightly more water soluble. And they're clearly both fairly small. They're on the small side. And the other thing we want to say about them is that they're always neutral. It's tempting sometimes for students to try to remove a proton here or here on that alcohol, but those types of alcohols have pKa's around 20 or so, maybe even a little bit higher. So we could make a note of that as a pKa greater than 20. And because of that, you can never get up higher than 20 for the pH, and therefore you can never deprotonate those. Moving on. The next one is cysteine. And cysteine is an awful lot like serine, except instead of having an alcohol, it has a thiol. And that is going to be about the same size as a serine. It's not a great hydrogen bond donor or acceptor, and it's not always neutral either. It can deprotonate. And if it did so, it would become a thiolate, S-. It doesn't do that until you get up to about 8.5. So we still consider it polar neutral because that's typically the way it's going to be, protonated and neutral at normal pH. Moving on, we have tyrosine. Tyrosine is a bulky aromatic. Sometimes people throw it into the same group as phenylalanine and tyrosine. Rather phenylalanine and tryptophan, because it does have a certain nonpolar character about it. But the presence of the alcohol on the phenyl ring means it's somewhat polar, and you can even have hydrogen bond donor and acceptor with that group. Interestingly enough, you can also make it deprotonate, but not till you get up to about over 10. And in that case, it would become a O-, and that's not something we typically see under normal neutral pH conditions. So this is all stuff that goes with tyrosine. And lastly, at the bottom of the page here, we have 2 that we think of as a group together. They're so similar to each other. Asparagine and glutamine, ASN and GLN for their 3-letter code. They have amide side chains, and make a note of that. Amides, they're either 1 carbon or 2 carbons out, but they behave chemically very similar to each other. They're a tremendous H-bond donor and acceptor, and of course that's going to be good for water solubility. Everything on the right-hand side here, everything on this page actually is good for water solubility because of that ability to interact with water. And one last thing is very, very important about this. These are always neutral, and that means cannot deprotonate. It's a fundamental fact about amides. You can't put a proton onto that to make NH3+, and you can't take away a proton to make NH-. Always, always neutral, and that's going to be tempting to try to change that, but we can't do it. So that's our polar neutrals, and we're going to continue on with the left-hand side of the page, the polar charged, either positive or negative, in the next video.